All right. So we're going to go back here to my whiteboard. Okay. And I just lost, when I left, I lost all of my drawings. Cute, 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 cute. Oh, I see what happened. Never mind. The reason why I lost it is because I'm not on the whiteboard. There we go. Now I'm back. Okay. All right. So proteins make these big, long strings. Okay. So remember, atoms are always moving. The atoms in the molecules are always moving. The molecules are moving. Okay. And that measurement of the movement, okay, the hotter it is, the faster they move. The colder it is, the slower they move. Okay, so temperature is a measurement of how fast the molecules are moving. Okay, so it's kind of cold outside today, but it's warmer than it has been. The sun is, the earth is tipping back toward the sun. We're getting more light, more heat, and so things are warming up. Okay. All right. So if I were to draw this guy, it would be this long string. Okay. What happens to string or yarn? Okay, when you just have it laying around in a big long string, what happens to it?
It gets tangled.
Tangled.
Yeah, it starts getting tangled. Okay. Or,
Mitchell.
sorry, what?
Oh, I called Jinx on Mitchell because we said it at the same time.
That's a good thing. Yes. Yeah. So now, if I'm talented, your cells are talented. Okay. Instead of just getting a random tangle. Okay. What they do is they fold these into interesting shapes. So it's kind of like your cell takes this string and knits it or crochets it into different shapes. Okay. But this string, the sequence of amino acids, we call the primary structure. Okay. In other words, it's the first structure that's formed. Okay. So it's kind of like the string of a yarn coming out of the factory. Okay. All right. So I'm going to clear this so I have room. Okay, so I'm going to draw my primary structure up here. Okay, so there are two secondary structures. Okay, so this string can start to fold into kind of a spring. Okay, now we call this an alpha helix because it was discovered first. Okay, so remember, on this string, on the nitrogen, I have a hydrogen hanging down, and on the carbon, I've got oxygens hanging down. Okay. So because this is bonded to a nitrogen, okay, and this is bonded to a carbon, these get a little bit of a positive charge. Okay, so these guys, I'm not going to do the delta because I didn't leave enough room. I get a little bit of a positive charge here. I get a little bit of a negative charge here, and they become attracted to each other. Okay, so I get hydrogen bonds that form between these H's and this O further down. Okay, and it stabilizes this structure. Okay. Now, depending upon what else is going on, okay, this tends to be very rigid this way, but it can stretch this way like a spring. Kaboing. So in flexible proteins, okay, we tend to find a lot of alpha helices. Okay. Now, another structure that can form, okay, let's say that I've got my string going back and forth, kind of like cloth. Okay. I still have these hydrogens and these oxygens that are attracted to each other. Okay. So we form hydrogen bonds between them. And I get a sheet going. Okay, and that stabilizes the protein. Now we call this a beta pleated sheet. Okay, so that is a part of the secondary structure, and they're all because of hydrogen bonds. Okay. Okay, now I can take this and I can fold it further. Okay, and so this is the first shape, this is the second shape, this is the third shape. But we use Latin, if I remember correctly. I'm thinking it's Latin. And we make a tertiary or third structure. Okay. So far it's been these guys that have been hydrogen bonding to each other. Okay. I still have these R groups hanging down. Okay. And these R groups are going to interact with each other. Okay. So I'm going to continue to fold things. Okay, so let's say I've got an alpha helix and I've got some beta pleated sheets, and I'm coming back around and doing all sorts of flippy fun things. Okay, if I have a side chain that has a negative charge, okay, and over here I've got a side chain with a positive charge, they're going to be attracted to each other. And it's going to help stabilize that shape. Okay. I can also form hydrogen bonds between my hydrophilic side chains. Okay. And then for my hydrophobic side chains, okay, I've got water outside here. Okay. And so the water is going to force the protein to keep its hydrophilic side chains on the center away from the water. Okay. Or later next week when we talk about lipid membranes, okay, we can, okay, so if I've got a lipid membrane, okay, I can take a protein, and if it has hydrophobic side chains, I can embed it in a membrane. Cool, right? So if it's like this and I've got water out here and water out here, these side chains are going to be hydrophilic, hydrophobic on the inside. Okay. And I can make all sorts of things. Okay. I can make most of the structures of the cell, and they do just about everything because I can fold or knit this string into any shape I want. Okay, how are we doing? Have I flipped your brains inside out yet?
Yeah, I kind of want to cry now.
Yeah, you know, when you're exposed to brand new information, you're exercising your brain kind of like a muscle. But you know, your brain isn't moving, but you are expending energy. And you know, you've studied enough when you get a headache. So it's kind of like when you're doing physical exercise and you get that burn in your muscles. It's your muscles saying, okay, we're done, we're done. So tell you what, oh, go ahead.
What kind of questions would you ask us then on the test? So, I mean, because this is a whole lot of what the hell, clear as mud. I don't know what the heck you're saying to me.
I know it's another language, and it's going to feel like that until you have enough intensive repetitions. Okay. So tell you what, we're going to take a minute break because your brain has had enough. So it's kind of like you're at the gym. You're getting off the treadmill, you're getting off of the weight machine. So think about something else. and then we'll do some quizzes, okay? All right, so I'm gonna stop share and let's see, come on, stop share. And I'm gonna go back to the PowerPoint and I'm going to advance to the point where we've got some questions, okay? And now I can't see you until I stop my, there we go. No, I do not want to keep those. Come on. Stretch, breathe deep. It's always good to take breaks when you're studying. Because otherwise your brain gets to the point where it says, okay, I'm done. I have had enough. And your mind wanders. And so it is perfectly normal when you're studying something you're not particularly interested in, or if it's brand new and you're spending a lot of time trying to get a handle around it, it is very good to take breaks. It's okay for your mind to wander, okay? Okay, are you ready? Are you ready to go back and answer a few questions? We've got about 5 minutes left, so I can't torture you too much. Okay, so I'm going to pull up a poll. And unfortunately, I do not have a 4-question poll yet, so ignore E, okay? So carbohydrates A, lipids are B, nucleic acids are C, proteins are D, okay? Here's my first one. Okay, table sugar, cane sugar, and beet sugar are all composed of sucrose. Sucrose is a oh, I didn't launch it. Sorry, you can't vote yet. There we go. Now I've launched it. Okay, we've got some good early voting. We passed 50%. Oh, we're past 60%. Good job. We're at 73. 80%. 86%. I need 2 more people. Okay, I'm going to go ahead and end the poll and share the results. Okay. Okay, so for the sake of time, I'm not going to do the tell me why you picked the answer you picked. Okay. The correct answer is carbohydrate. Okay, because sugars are carbohydrates. If you picked lipid or nucleic acid, that's just fine. We're still learning this. This is still a foreign language. Okay. And forgetting is a part of learning. Okay. Your brain dumps new information. Okay. And so now you're going to remember it for longer. Okay. All right. So let's get rid of this. I'm going to stop sharing. And I'm going to zip past the questions that I had here. Okay. And we're going to go to a different one. Okay. Oh, let's skip the functions. You can read about that in the textbook. Those are pretty straightforward. Okay, so here is a structural formula. Okay, so I'm going to bring the poll back up and I'm going to have you vote as to whether that is a carbohydrate, a lipid, a nucleic acid, or a protein. Okay, we've got some good early voting. And remember, it's okay if you get it wrong, especially at this point. This is practice. Okay, this one's not as easy as the table sugar, beet sugar, cane sugar. Okay, we're at 83%. I'm going to go ahead and end the poll and share results. Okay. Okay, we were pretty close to being even. Okay. 2 for carbohydrates, 4 for lipids, 4 for nucleic acid, and 3 for proteins. Okay. And that is just fine at this point. Okay. If you keep working at it and keep quizzing yourself, okay, eventually you're going to remember. Okay. This is actually cholesterol. Okay. And so that makes it a lipid. Okay. Let's see. Let's do one more. Let's do one more just for the fun of it. You're going, this is not fun. Okay, so I'm going to get rid of this one and pull up that one. Okay, is it A, a carbohydrate, B, a lipid, C, a nucleic acid, or D, a protein? Okay, we've got some good early voting. Ooh, we've got a tie so far. Let's see if we can break the tie. Okay, good job. Okay. So it was pretty evenly split between carbohydrate and nucleic acid, okay, which is really very good, okay, because remember nucleic acids have that sugar group, okay, but this doesn't have a phosphate group and it doesn't have a nitrogenous base. I know this looks kind of like a base, but it doesn't have any ends, okay, so it's a carbohydrate. Okay. All right. So I do have a practice quiz that I wrote that you can go through and practice that. Also, the dynamic study is a good way to study this. Also, going into what to do for next time. Come on. If you start the biochemistry badge, this is another way to have some good repetitions, okay? And believe it or not, right now, it's like I'm speaking Klingon. None of this makes any sense, okay? And when it's brand new information, you need about 6 intensive repetitions. I'm sorry. It's unlike the Matrix. You can't just download this and keep it, okay? So that's why I have so many ways for you to study this material, okay? So the biochemistry badge is not due this week. It's not due tomorrow. And the case study one is not due tomorrow, okay? But the case study is due a week from tomorrow. The biochemistry badge is not due for about another 3 weeks, okay? But if you work on it and continue to study it, it's going to start making more sense. It's like when you're learning a language, okay? Is that reassuring? Have I reassured you, or are we feeling overwhelmed? Okay.
Overwhelmed, definitely overwhelmed.
And you know what? Overwhelmed is a normal response. It's a normal response. And you are not the only one. So if you feel overwhelmed, go ahead and take a little bit of a break. Okay, don't worry about it for the rest of today. Okay, go ahead and work on the library badge, okay, which has nothing to do with chemistry. Okay, give yourself a little bit of a break and then come back to it. And eventually it'll feel less overwhelming. Okay, eventually the old light bulb will go on overhead. I say old light bulb because we draw the Edison light bulbs as opposed to, I don't know, instead of LEDs. Okay. Also, I do have office hours tomorrow from 1:30 to 2:30. Feel free to drop in. I'm happy to go over this again. I'm happy to help. And Rochelle is happy to help too. Rochelle is really good at this chemistry stuff. Okay.
She changed her hours, right?
She did. Yeah, we weren't getting a lot of people earlier in the week. So her hours are 1 to 2, Thursday to Friday. Okay.
So basically during the same time as yours then, tomorrow.
Yeah, but sometimes it's good to hear how somebody else explains it. Okay. So if you've heard enough from me, Rochelle can explain it a different way, which is helpful. And then
And that
Rochelle is also, oh, go ahead.
And that counts towards our embedded tutor assignment if we do it tomorrow, right?
It does. It definitely does. Yep. And if those times don't work for you, you can email me and I can set up a different time that'll work for both of us. Or you can post a question to the discussion board and Rochelle will help you out that way.
But if we meet with you, then that doesn't count because we have to meet with Rochelle for it to count. Is that correct?
Yes. Although the first time you meet with me, I will give you 3 points of extra credit. So meeting with Rochelle is required. Meeting with me is optional. Because you already meet with me twice a week. Okay, any other questions?
I have one, but they're from the beginning of class. I just wrote them down. So
Oh, okay.
you're talking about hydrocarbons, and
Yes.
my Zoom decided to update, so it kicked me out of the meeting.
Of course.
So what made it hydrophobic?
So it was hydrophobic. Let me go back to the whiteboard and I'll show you that. Okay, so let me get rid of this. Okay, so because hydrocarbons, excuse me, yeah. Hydrocarbons, they are carbons with hydrogens on them. All of a sudden I was confusing myself with carbohydrates and hydrocarbons. They are very similar. Carbons are close enough to this, they're smaller than oxygen, so they don't pull on the electrons as much. Okay. And so these bonds are non-polar covalent. Okay. So the electrons that this hydrogen and this carbon are sharing spend equal amounts of time between the carbon and the hydrogen. And for this one, it does the same thing. Okay. And so we don't get that partial charge. Okay. So instead of, like, if I have water over here, because the electrons are spending more time around the oxygen, we get a little bit of a negative charge. We get a little bit of a positive charge over here. Okay. Over here, instead, we have no charge. So they can't form hydrogen bonds. Yeah. But let's go ahead and get an amino acid over here just for the fun of it. Let's draw it this way. I'm going to draw it on its side. Okay. So what happens with an amino acid? And I'm not enough of a physical chemist to know why this happens. With this, the hydrogen will leave this and I'll get a negative charge here. But that hydrogen sometimes will come over here and this nitrogen will get a positive charge. Okay. Whoops, I'm erasing my stuff. So let's put these guys back. So because I've got a positive charge and a positive negative charge there, okay, I can form a hydrogen bond here, okay, and for my water, okay, let's say I've got a water here, okay, I can form a hydrogen bond, okay, or an electrostatic attraction between this negative charge here and this positive charge here, okay. So that's why amino acids are hydrophilic, okay. It's because we've got oxygen involved and we've got nitrogen involved. But carbon doesn't pull on them enough. Okay, did that answer your question, Mitchell?
Yeah.
Okay,
And then our
other question. Oh, go ahead.
Our 2 assignments this week. Do we only have 2 assignments this week?
Just one.
Just
The
one?
library badge.
Okay.
But I do encourage you to start the biochemistry badge. Once you finish the library badge, I encourage you to start the biochemistry badge, the biology chemistry STEM badge, and also start the case study number 1, because that's got some chemistry going on in it too. Yeah, chemistry everywhere.
So you're saying that Rochelle cut her hours because there wasn't enough participation, but it was pretty easy to begin with. So, but now that we're getting
Right.
into the harder stuff, will she add those hours? So then that way
She
we can get
might.
that. Okay.
Sure. I'm going to be meeting with her tomorrow, and I'll see if, especially for this section, if
Because this is clear as mud.
Yeah, it is. Chemistry is hard. Chemistry is hard, especially the first, for me, the first couple or 3 times I went through it. It took about 3 classes before I went, oh, okay, I'm getting it. Oh, here's Rochelle. She's not opposed to adding more hours. It just seems most people prefer the discussion board. Yeah, so Rochelle, since you're still here, what would you say to tell you what?